So tensors are mathematical objects that are very useful for capturing data. Imagine that our data first is given by genes, so we can collect all the information with respect to genes in an array or what we call a vector. Then say that you add to your information, you add a second parameter, which is, for instance, cell types. And now, for each cell type, you have all the genes that you are considering, and so you can encapsulate all this information in a two-dimensional table, which we call matrix. And now say that this is the information with respect just one sample, but you have, for instance, two samples, and now you want to consider them at once, so not separately. And so what you do is you have the first matrix with respect to the first sample, and then you consider the second matrix with respect to the second sample, and you literally stick them together, forming a three-dimensional cube. So you have the two slices, the first sample, second samples, and you look at them like two slices of a cube. And this is a tensor. This is a three-dimensional tensor because it depends on three parameters, where the third one is samples. But you can do the same with a higher number of parameters and you would get multidimensional tensors. So now we have a nice way to collect our data, but now we want also some technique, some way to analyze this data, to study this tensor. And for instance, for what we can do for matrices, we start with a matrix M, and there is a way to write it in a more expressive, more descriptive form, which is called principal component analysis, or also singular value decomposition. And more precisely, you rewrite M as the product between three matrices, U, sigma, and V, where sigma has the same size as M, and has the very beautiful form, which is diagonal. So all entries of the matrix are 0 except the one on the diagonal, which are certain values, d1, d2. And we can also add the extra information that d1 is greater or equal than d2, greater or equal than d3, and so on and so forth. And we stress out that we are not changing our matrix M. We are not altering our information. We are just rewriting it, reorganizing it in a different way. And in particular, what we can deduce from this form is that the values on the diagonal, which are called the singular values of the matrix M, are related to the most identifying feature of our data. And in particular, disordering means that the feature related to the first principal component, the one on top left, is the most identifying feature. And okay, this is also useful if we want to approximate our matrix M. In this case, it will be enough to set to 0 the least value, and this would lead to the best approximation of M. We can generalize this for matrix to tensor, to higher order tensor, and the technique is called HOSVD, higher order singular value decomposition. Now, instead of starting from a matrix, we start from a tensor, let's say a three-dimensional tensor, and we want to apply PCA. So the first thing that we do is we project our three-dimensional tensor in order to get some matrices, and we do so, we project with respect each parameter. So we will get three projections, and these projections can be understood in this way. So here we have a three-dimensional tensor, depending on gene, cell type, and samples. We have the two matrices with respect to the two samples, and now we unstick them and then glue together. In this way, we get a matrix, which is given by the matrix with respect to the first sample, the matrix with respect to the second sample. And here the rows will be indexed with respect cell types, and instead the columns with respect genes and samples. And now that we know how to project, we do this with respect to each parameter, to each dimension, and now to the single projection we apply the PCA, and so we get a decomposition of this projection as U2 times sigma 2 product V2, and we do this for each projection, and then we collect at once all this decomposition, and HOSVD allows us to rewrite our original tensor as U1, U2, U3, given by, these are information given by PCA, applied times another tensor, which still has the same dimensions, the same number of parameters than the former one, but now as a more descriptive structure. Similarly to what was done for matrices, now this won't be necessarily diagonal, but still a more descriptive structure. And so, to recap, what we did was starting from a tensor, which contains a huge amount of genes, and in order to study, to analyze its feature, with HOSVD, we reorganize its structure in order to get a nicer tensor to deal with.